All right, well, welcome everyone to this year's ClinGen Clinical Genomics Career Panel Series. Today's panel is focused on research, development, and other opportunities. We have a wonderful lineup of professionals today who will be telling you about their training and career paths. Just a few housekeeping slides to get us started. So as a reminder, this is a 5-week series that we are putting on every Tuesday. We are currently on our 4th panel. So today, again, focusing on research, development, and other opportunities. Next Tuesday, we'll be focusing on stepping stone opportunities. So these are roles that help individuals gain experience and knowledge before further education and jobs in the clinical genomics space. That will be next week. And our 3 previous panels focused on genetic counselors, clinical lab opportunities, and physician clinicians are all available on our website right now as recordings. So you can access both past and future recordings on the website. And to get us all started today, our panelists would like to know a bit more about you, our guests, before we get started. So a poll is going to show up on your Zoom window, and we just ask you to let us know if you will be a student as of September 2022. And the second question is, how familiar are you with research and development career paths? So we'll give everyone a moment to respond there. Okay, and you will see we have the results of the poll. So it looks like the majority of our guests today will not be students in the fall, although we do have a fair number of undergraduate students. So a variety of attendees on the call today, learning about different career paths. And as far as familiarity, it sounds like most people on the call are slightly familiar or moderately familiar with research and development opportunities in clinical genomics. So we'll keep that in mind as we go through the panel. A few more notes for you. You will have the ability to ask questions throughout the panel. We do ask you to use the Q&A box rather than the chat feature. So if you enter in the question and answer box, we'll be able to see those questions and ask them of our panelists. And you can also upvote. So if you see something that you would like to ask has already been addressed, please let us know you would like to see that one asked and we'll prioritize it. All right. Oh, I did everything out of order. So I did our poll before, but here we are. So finally, we can get started and introduce our panelists who will be speaking with us today. I am going to list everyone in the order of their slides just to give a brief overview of their training and their current role. They'll have more opportunity to talk throughout about what they do. And then I'll ask each of the panelists if someone talks about a similar path that you've taken, feel free to just defer and say you took a similar path to someone who spoke before you. So with that, I will turn things over to Dr. Nara Sabrera to introduce herself.
Hello, everybody. Good afternoon. Thank you, the organizers, for having me here this year again. It's a pleasure. I am a clinical geneticist and a researcher, and I work at Johns Hopkins here in Baltimore, US. And I'm from Brazil. So I did medical school in Brazil. I did a residency in clinical genetics after that. And then I came to US to do a PhD. And my intent was to go back to Brazil. But then, as I was working here on my PhD, I decided that I maybe would like to stay, but to do medical career here, I needed to take these steps. So towards the end of my PhD, I started working for the steps. And then when I finished the PhD, I did a short postdoc while I was finishing my steps to apply for residency in clinical genetics again, because to do clinical genetics, even I had done the steps, I needed to do residency here again in clinical genetics. So I did a 2 years fellowship residency in clinical genetics, that all at Hopkins, and never went anywhere else. It was already too much work. to do it all. I would not like to be looking for other places to go. So I stayed there, did my clinical genetics residency. And then in 2015, I started as a faculty and I have a research lab and I do 80% of research and 20% of my time in clinic seeing patients with rare Mendelian disorders. I think that's all I need to say, but if there's anything else that you want to know later, let me know.
Excellent. Thank you so much. Next, Dr. Orion Buskie.
Oh, thanks, Danielle. And thanks so much for having me back this year as well. I am the CEO at a company startup called Phenotypes. So we're a small Canadian startup making software in the clinical genetics space. My background, I guess I am actually originally from the US, but now based in Toronto, along with most of our team. Have been interested in genetics for a very long time, from back even in high school, and then ended up doing an undergrad in Washington State in bioengineering and computer science. Took basically a year after that to work at a research group to figure out next steps and to plan sort of applying for graduate school. I worked on the ENCODE project during that time and sort of got in the mind space of working on large scale genomics and collaborative research projects. And eventually I wanted to teach and do research in academia. So I went the PhD route, ended up talking to a bunch of different people and interviewed at a number of graduate programs and ended up finding a really good fit here in Toronto at the University of Toronto computer science program. So I came here, started working on computational biology and algorithms specifically for sort of genotype, phenotype, and rare disease patient matchmaking, started working on matchmaking platform and software and algorithms around that and the matchmaker exchange, and got into sort of standard setting and standardized data with the Global Alliance for Genomics and Health. So through those projects, got to meet a number of the other panelists over the years. Yeah, and I think sort of just to round things out, what I'd say is that during that time, and as I was looking at a lot of other people and mentors, a lot of the professors that I found to be the most compelling were those who had some experience in industry or experience outside of academia. And so I was really interested in translational projects and getting sort of my hands on things outside of academia. So wanted to move into entrepreneurship, and so came and joined Phenotypes, which is this spin out of the research group that I was in. So I've been here for 5 years now. And yeah, going through all of the fun things associated with that in terms of raising venture capital and growing a team and all of that sort of stuff.
Well, glad to have you. And next up, we have a Dr. a software programmer, very importantly, Neetu Shah.
Hi, hello everyone. I'm happy to be here and thank you for having me. It's very good to see you all again this year again. Yeah, I am a manager for software programming and engineering at Bioinformatics Research Laboratory led by Dr. Alex Milosavljevic at Baylor College of Medicine. And I'm originally from India, and when I was in India, I did my master's in biology, actually microbiology. And it's like where I was working in a lab there in India that was funded by Indian Council of Agricultural Research, and they were doing sequencing, and I got into bioinformatics. And when I came to US, I went to a graduate school and did my master's in computer science. So it's kind of a big turn for me from biology to pure computer science. And when I joined Alex's lab, I started as a programmer, both as a backend and front-end programmer, more than a bioinformatician. So that's what I wanted to do. I'm more and more got attracted to the pure computer science and building applications. And I have been with Alex for the past 8.5 years, right when ClinGen started, actually. And I'm right now, I manage a couple of products. I am a lead engineer of a couple of products. I'm not sure whether you're familiar with it. ClinGen's evidence repository, actionability.
So yeah, I'm a biocurator and ontology developer for the Translational and Integrative Sciences Lab at the University of Colorado. We're at the Anschutz Medical Campus in Aurora, Colorado, outside of Denver, and I work remotely from Portland, Oregon. So my undergraduate training was in biology at Reed College in Portland. And then I did a PhD in cell biology at Oregon Health and Science University, also in Portland, where I was a bench researcher working on a mouse model for cancer immunotherapy. And I didn't want to stay on the bench and was looking for what everyone described as alternative career paths. I did do a short postdoc in cancer research and taught a little bit at the community college here in Portland. And then I joined my group at the TIS Lab in 2010 as a biocurator. And we build data standards for annotating rare disease data, specifically focusing on disease to phenotype annotations. And I work a lot on developing phenotype and disease ontologies and doing curation of data.
Thank you so much for that introduction. And Adriana, if you could just do your introduction briefly, and then we'll get to our discussion as if we were never interrupted.
Right. Hi, everybody. So I am a genetic counselor by training. I, as many others on the panel, was not born in the US either. I am from Portugal, but I did my undergrad in New York and my graduate school, the genetic counseling in DC. And I currently work at the NIH, the National Institutes of Health, specifically at NCBI. I will spell out the acronyms because we love these acronyms, right? So at the National Center for Biotechnology Information, and I lead 3 different projects. So I lead a project that's called MedGen that has to do, it's a website for information about clinical genetics. One of them, it's called GTR, or the Genetic Testing Registry, and that has to do with data submission about like test descriptions from laboratories that actually provide the genetic tests, and also the medical genetic summaries, which is a resource specifically like content that we create specifically for pharmacogenetics. So that is my role at the at NCBI, and it has been my role since, well, I joined in 2010 and then progressed into the leadership position that I currently hold.
Wonderful. Well, we thank you all for joining us today. Just going to do a refresher that today's career panel brought to you by ClinGen is focused on clinical genomics research, development, and other opportunities. We're joined by 5 wonderful panelists who work in clinical genomics in different areas of expertise, and we can dive into some questions today about what they like, how they got to their roles, their training. I'll remind everyone to please put any questions that you have into the question and answer feature on Zoom. And I think I'll get everyone started with a question, just for anyone who wants to answer. How did you hear about your career?
I guess I'll go first. So genetic counseling, into like I'll go with genetic counseling and then I guess my actual career. But genetic counseling was something that actually when I was in undergrad, that was not something I was familiar with. I just really liked genetics. And it was very like serendipitous, I guess, like one of the other students in one of our developmental genetics class was talking about genetic counseling and how she had gotten accepted. She was a year ahead of me, and how she got accepted to this thing. And I'm like, wow, that seems actually pretty cool for me, because I love genetics, but I didn't want to do just wet lab. Like I kind of like the patient-facing part of genetics. So that's kind of how I even was introduced to a brand new profession for me that I just wasn't aware existed. And that's why I decided to go to graduate school for genetic counseling. And the beginning of my career was actually in a pediatrics clinic and a more traditional role in genetics. And then I saw this opportunity at the NIH, and NCBI was something that, like, I mean, I used some of the resources, like variation viewer and things like that. That was like for me, like, these things are valuable, but it takes a really long time for me to like even understand how to use them, right? Like at that time. So I think in CB, I kind of realized that, right? Like we have all of this information and this amazing resources, but we kind of need the clinician side of things. So they hired, at that time they hired 2 genetic counselors and then a medical geneticist after that, to kind of bring that perspective. Like, yes, we have all of this data, but we need to make it useful not just for the researchers, but also for the clinical community. We want people to be able to access this. So they brought us in to kind of bring that perspective of turning all of this wealth of information into being useful in the clinical worlds.
Mine was a little easy.
Yes.
My father is a clinical geneticist and researcher, so that was a easy exposure, and then I liked it. But I didn't know if I would like medical school. I always knew, or I thought I wanted to do the research side. So when I went to medical school, it was kind of a lucky thing that I really liked the medical school. And now I kind of say I like more the patient side than the wet lab side, but I still love the research part. So I do both, but was lucky that I actually liked medical school because I was thinking more on the research side of the things when I started.
For me, I didn't know what biocuration was. And I, like I said, I was looking for alternative careers. I wanted to leave the bench when I was in graduate school. And I actually went on tons of informational interviews per the advice of the career office that I went to. And I talked to so many people in different fields and never talked to anyone doing biocuration or ontology development, and ended up just applying for open position at Oregon Health and Science University, OHSU, where I went to school, and ended up getting hired. And even when I was hired, I was still like, I'm not really sure what this job is. And then it turned out to be a perfect fit and the perfect career path for me. But the whole field of biocuration is kind of small and still relatively new. So I hope that through these career panels too, we can introduce more people to this career path, because it's really quite fun. And I think so that there's not a lot of knowledge, or it's not as well known too. So yeah.
Well, I'll throw something out there for all of you. What do you like best about your current role? Who would like to start?
I was waiting for the people who didn't talk to talk. So Orion and need to.
You want what?
Talking all the time. I'll let you both start with that one.
Oh, goodness. I think I had a hard time with this question because I am having trouble defining what my career is. It is a collection of various different projects, is kind of the way that I see it. But ultimately, in sort of the current role and in entrepreneurship these days, it's all, every day is learning something new, which is really, exciting and engaging. So yeah, no, it's great on that front.
Lifelong learning.
I really like the problem solving part of my career. I really like that. Maybe that's the main reason I went into core engineering. I really like building things and breaking too. But now I enjoy more keeping in pace, because it's very fast. The technology keeps changing. The solution that was good 2 years ago is not efficient anymore. We have to rebuild it and update. And so yeah, mainly problem solving is really the key for me.
I would say the novelty, like Orion was saying, but on my clinic, I'm always seeing a patient with a different disease. It's never the same. So you don't see, you go to a diabetes clinic, you are going to see patients with diabetes all day long. In our clinic, in clinical genetics, every patient that comes through the door has a different disease, and it's always a puzzle. You have to like put the pieces together and try to discover what the diagnosis is. Sometimes it's the only patient that you will see with that disease in your whole life. So, and then actually the research part is the same, always trying to discover what is the genetic basis of these diseases that are very rare. So it's always a novelty, and nowadays the technology is going so fast. There's always a new method, a new way to do things. So I think that I like that very much also.
I like in our group too, we collaborate with a lot of people and do a lot of team science. So I like all the collaborations, and I also like the ability to work from home and work remotely, but we travel and see each other at conferences or have project meetings, which has been much less during COVID, but starting to get back into it. And it's nice to interact with so many people and on such different levels too. It's really fun.
Okay, so I think I'm the one left, right? To do something different, you know, like a non-traditional, like I'm not, you know, there was some activation energy with talking with, like, you know, developers, and it's, you have to learn a different language, and that alone was kind of exciting. But it really is the whole idea that coming from a clinic and coming to this role, being made aware that, like, oh yeah, everything that you use on the website that looks so seamless, right? And it's not, you know, since our teams have to have experts in, you know, like you have developers and web designers, and there's a lot of people involved, but bringing the perspective of, like, yeah, that's nice and all, but like, you know, like bringing that perspective of, like, you don't have half an hour to figure out, make that part, you know, like easier, or just kind of being able to world, the clinical genetics, the current needs, and then bringing that into the, hopefully, the resources that people are using, making sure, you know, like we talk with Nicole quite a bunch, you know, like these kinds of collaborations that need to happen in order for the person that's then, like, with the patient to, you know, have easy access to information that's actually accurate and up to date and, you know, makes sense, and we can help them on that end. But, you know, like having that input of, like, yeah, but then all of this behind the scenes work has to happen in order for the front end to, you know, for you to be able to really help your patients. And you're exposed to this, like, I mean, medical genetics is just such a huge field, right? So trying to make that in a way that any one particular clinician can, you know, have access to everything they need, that challenge is kind of fun for me.
Well, thank you all. We do have a question from the audience. How do you see genomics changing in the coming years, and how does your role support those advances?
It's changing all the time. In the last 20 years, it has changed so much. And I think understanding the genome is opening the doors for many more things, right? Understanding the genomes and the variation in our genome is really allowing us to do better diagnosis, to look for better treatments, to do better counseling for the families. And I think it will just get better. We are having more and more tools that will make the genome, what we know about the genome, more precise, and that will translate, I hope, as it has in the last years, translated. And I think particularly what I do is try to connect what we know about the genome, our genomes, the human genome, to the disease that we see in clinic. So I think that's how we in our field, in the clinical side, are trying to help the understanding of the genome, how these are connected to the diseases that we see. But I think it's going to change to better as we understand better the genome. We can care more for the patients with disease, not only rare disease, but complex disease also. It's not only one kind of disease or the other. It's for the whole field, I think, of medicine.
The work that our group does is developing these standards for standardizing data. So we actually just put out a paper called about phenopackets, which is a standard for describing patient phenotypes, and then that can be used for comparisons amongst patients, and then also with helping. We also work on the Monarch Initiative, which compares phenotypes in human patients to animal models as well. So the phenopackets are a new standard that hopefully will enable comparison of phenotypes amongst patients and against model organisms and help with identification of variants and genetic alterations that are seen in some of these diseases. So I think the advances will help us. I think we'll start to see better diagnosis of patients and identification of diseases. I work on a standard too, that the Mondo disease ontology, which describes different diseases, and we get a lot of requests for new diseases to be added to Mondo all the time. So I think we'll start to see more and more diseases get actually defined and identified, and hopefully improve diagnoses and then treatments for patients.
I think there are two big trends that are pushing things. One is the rapid increase in the availability of genetic testing as part of sort of clinical practice. So sort of mainstreaming what in a lot of places is mainstreaming, where it's earlier and earlier into the clinical process, genetic tests are being ordered, and they're being ordered more and more clinically rather than necessarily funded through research projects. So where this exome and genome sequencing is becoming so common, and then based on sort of all of the research findings and improving Nara and Nicole, the knowledge bases that are being built up and the data standards that everything is now sort of running on are sort of this framework that are allowing software tools and allowing the processes so that we can diagnose people that much faster and that much more accurately as part of sort of mainstream genomic medicine. I think that's sort of at least the vision that we're seeing about how this field is evolving over the next 10 years, and where sort of software meets data standards, meets clinicians and genomic specialists playing roles in providing that.
Thank you all for weighing in there. We do have another question from the audience about a typical day in your work life, if you have a typical day, or you could take the option to do, what does a typical project look like when you talk about working on a project? How long does that last? How is that set up? So we'll throw that out there for our next conversation.
At least I can start that.
Yes,
at least in my field, there isn't a standard day for me. It's very different. It depends on what you're working on currently. If it's a new product, then at this point in my career, I'm a lead engineer, a lead designer as well. So it starts with designing. Sometimes I'm full-time coding. Sometimes I'm reaching out and managing the team. Right now I'm a manager too, so I have a team that works with me. So it's always exciting, and it's never the same a single day. And meetings, of course, that's new. For the past 2 years, I have been attending a lot of meetings too. So yeah, it's very exciting, and it's never do this A, B, C, and every day it's never boring.
I would say something. When you're on the research side, there's something that you don't think a lot before you go into the research. That is how much you have to write. So get ready if you want to do research. Because I never thought I would have to write so much in my life. So you have to be writing papers, you have to be writing grants. And that is a big part of keeping your research going, and because you need funding, and to have funding to get your research going, you have to be writing papers and writing grants. So, and then you have to also be very patient and very resilient, because you're going to be getting a lot of bad reviews about what you write. Right? So like, doesn't matter how I have heard it from everybody. Doesn't matter how senior you are and how talented you are and how successful you are, you will get reviewers saying that what you're proposing is not good enough, or what you just wrote is not good enough, and that you should change here or change there. So be ready for these two parts. Like they can be fun. It can be a learning process when you're back and forth trying to get a paper accepted or a grant funded. But you have to be ready for that. So it can be more learning than frustration. So that's what I would say.
I'd say working in academia, I also, in addition to writing, give a lot of presentations and go to a lot of conferences, and also do a lot of service types of responsibilities. I'm on a lot of different committees. Like I'm on the our professional society, the International Society for Biocreation. I'm on the executive committee for that. I've been on a lot of conference organizing committees. I just got back from a conference where I was one of the organizers. So, but yeah, my typical day-to-day is pretty dynamic, which is fun. I usually start around 8 in the morning every day and have conference calls right away. I'm on a lot of conference calls. But I think especially working in academia, I'm not sure how this is for everyone else, but I feel like I have a lot of flexibility and I get to kind of decide how I'm going to manage my day and how I'm going to miss outside of the calls that are scheduled. I get to kind of prioritize how I want to organize my projects and how when I get my work done. So that's a really nice aspect of it.
And that's very important to people who want to have children, Nicole, and have a career. The flexibility that we have with academia, it's very helpful when you have a young family and you have children. So that's another thing.
So I guess I'll go next. So the world that I have right now, it implies different groups, like different teams within NCBI. So we have the development team, and you have to adapt. For example, we went through this change on how they work. Like there's this big push on becoming more efficient, and as you're dealing with like more data, and you have to develop more like tools that are more efficient to handle the increase in data, for example. You have to make some changes that are quite technical, that like you have this relationship with the development team, but also the development team itself, like how they work and how they produce their work. They also went like through this change, like I don't know, in where we work, like we adopted the agile workflow to become more efficient in how they do their own work. So coming into like a non-traditional role, you have to keep in mind that like it's not just going to be like about the genetics. I mean, I'm a genetic counselor. Like you have to really step outside your comfort zone and learn about many other things and like dealing with developers. Like it is a different world, but then you do have to adapt and see how they work and make sure that you can then like have a like your input is incorporated into how they work. And that's one facet of what I do, because the other one has to do with the actual content, with the data, with the like what makes sense to the clinician and things like that. So we call that group curators. It's just what we call in our at NCBI. And these are the people that either help the data submitters, like the labs that are describing their tests. They are the people that are actually curating the data, like the disease data, making sure that it's accurate and that everything makes sense and is up to date. And we put some value, right, like on that quality, the content quality. So that's the other facet. And then there's like a lot of collaborations that need to happen to, because one of the changes that is that because the field keeps on growing, the number of people working on certain parts of even genetics like keeps on growing. There's like not just genetics, in medicine in general, that you have to collaborate with. There's more groups that 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 you need to collaborate with, and then it becomes really important for the for data to be interchangeable, like for all of the all of the ontologies. Everybody has to kind of talk to each other, so it becomes really, really important for these networks and these collaborations to happen in order for for the whole of really medicine to benefit. And then there's that whole idea of like outreach and education, right? Like you can do all of this work all you want, but unless people hear about the stuff that you're doing and how to use it and how to use it efficiently, if they don't use it, then you're not doing, like you're doing work for nothing, which you don't want, right? So then there's also that push, like going to conferences, it's about the collaboration, but also about educating the community about the resources that are out there to facilitate the work that they do. So because there's all of these kind of things that you have to deal with, like a day is never always the same. And even if your meetings, like, you have a meeting every Monday at 11, but the discussions are always going to be different. That's kind of it for me.
Well, thank you very much. Thinking about kind of taking this in a different direction, we have our attendees on the call today who are interested in your career paths. Either from your own experience or from where you are now seeing others, what would you recommend for someone who wanted to pursue a similar career to yours before they go into a training program? Just have an opportunity to either volunteer or intern or take a job to gain some exposure in the fields that you work. Any recommendations?
Advice that I was given when I was looking to start my career is to go on informational interviews. And I thought that was really, really helpful. And people were so responsive. I would just cold call people basically and say, oh, I found you on LinkedIn or something and interested in this potential career path. Can I meet you for coffee? And then they would say that we would meet and they'd tell me about it. And then they'd say, oh, I know someone, and then introduce me to the next person. And I talked to so many people and it was really fun. And it was really, really valuable. I also recommend, especially if you wanted to go into something like I do, where we do a lot of open source software development, is make yourself visible. You could create an account on GitHub. You can contribute to open source projects. You can, if you have the opportunity to go to conferences, which is even easier now with a lot of them being virtual, but just meet people and make yourself known. Have an online presence. Create like a website about yourself and be searchable by Google in positive ways. Yeah, just to make sure people know who you are. And
actually just added, we just got another comment that's relevant to gaining experience, but also the ISB, which is the International Society of Biocurators. Is that right? Yeah. And can someone join? Do they have to be working professionally as a curator?
Oh, no, you can definitely join. So the Biocuration Society, the objective is basically just to promote the field of biocuration and to provide opportunities for networking. We do have a job board, so we list job opportunities that are coming up. We have conferences. We have a virtual conference that has been in 3 parts this year, and the final session will be on October 7th. So that'll be a great opportunity for anyone interested in biocuration to come. It's free too, so just join our free virtual conference and listen and hear about what's going on. Yeah, we have a website, biocurator.org, where you can find more information, or feel free to follow up with me individually.
Thank you. Right. Other recommendations for gaining exposure in your fields or on your career path?
Definitely going to the meetings. I think to the meetings on the field that you are interested on, it helps a lot, because then you can hear what's going on, you can see if it's really what you're interested on, and you can meet people and talk to people, and don't be afraid to talk to people. I was just having lunch with now an undergrad who came to this meeting where I am now as a high school student, and then she contacted me and did a summer rotation with me after that as a high school student, and she was on and off for almost 2 years as she could, working different projects with me, and now she's doing her college and she's here back in the meeting again. So like, and I was just telling her that now she is also doing a summer job in one lab. She already has a job lined up for the fall in another lab. exposure and talk to as many people as you want or as you can to get to know them and to get them to know you. So it will be easier for you when you're applying for things, and also to see if this is your interest, your real interest. I started going to the genetics meetings when I was in medical school, and we didn't even had genetics in my medical school, but I knew I liked that. So I was going to the meetings to meet people and see what I was going to, how I could get more exposed to it.
Yeah, I'm gonna second that whole, like, don't hesitate or don't be shy about reaching out to people. Thankfully, people in our field tend to be pretty open, and if we can help, I don't think anyone is, honestly, like, people are pretty open to helping out. The other thing I will say, some organizations, like where I work, right, like for the NIH, there are options for, or possibilities for internships or fellowships, depending on where you are in your career. And maybe if you're, a high school student, you could do a summer internship in like one of the areas that you're interested in. And that's, like, that goes a little bit beyond than just a conversation, like that really exposes you to whatever the job is that you're considering. And then when you have that real experience, it's easier for you to know if it's a fit or not, if you're actually doing it for some time. But getting your foot in the door, talking with somebody, like reaching out to people, is definitely a good way to go. And going to meetings, if you have the opportunity, is another really good idea.
Yeah, I think I would echo all of the great suggestions that people have just been sharing. And I think from coming, there are tons of ways to learn about sort of, or discover either sort of projects or areas of research or activity, whether it's companies or whether it's research groups or other things that are working in an area that you find, or working on a problem that you find really compelling. And I think once you've sort of started to, it's so much a process of finding out what you are interested or passionate about. And then once you've kind of started to find that, then I think the people in that space, or the research group or the lab or the companies that are working in that space, you can sort of engage with any of them, and there are opportunities for internships or research assistantships or co-ops, depending on sort of what the dynamics are. But there are always ways to start working or start getting experience sort of in that particular area, and then from there, sort of the sky's the limit. But I think certainly for us, like as a startup, we are constantly bringing on sort of interns and other students at different levels, and that was true for the research group that we spun out of as well. There was a constant flow of people, great people coming out of undergrad or high school who are, it was clear that they were interested and passionate about solving problems in this sort of space. And so then there's an opportunity for them to contribute and get experience. And it's a great win-win.
And one more thing that I forgot to say, like, so I don't know how many other organizations do this, but at least for the NCBI, we put together in certain topics, you can go into, like different organizations' websites, and NCBI being one of them that offers these things. And you can try, one or more, like as many hackathons as you want, but you're investing the time, but you're also like going through exercises and, like really getting exposed to whatever the area is that you're picking. But it could be a good introduction to see, like, yeah, this is actually something I want to go, deeper into, like, kind of like a initial exposure. That's another thing to consider.
Hackathons are great.
Right, we have another +1 for hackathons. In our final few minutes, maybe one or two responses from our panelists about a question that came in. Are there any misconceptions about either the role you hold or the setting you're in, such as research? Any misconceptions you can think of you'd like to clear up?
Okay, I don't know about misconceptions, but really quickly, I would just say, like, whatever you think that the role is, keep an open mind. From my experience, I came into NCBI thinking like, oh yeah, you want to see how a genetic counselor is going to use, blah, blah, blah site. Like I said, I had to learn how to talk with developers. I had to learn how to translate what's, my experience or what's in my brain into a way that someone with like very limited biology background needs to work. But to me, I took it as a challenge and it became actually kind of fun, but it's, you either have, like, you have to kind of keep an open mind. Before, like, I mean, the field is changing, right? So your position today is not going to be your position 5 years from now, like just out of like, the explosion of knowledge that we have. So I don't know about an open mind. If you're into the challenge of learning new skills, like it really can help you develop professionally if that's the path that you want to take.
Yeah, I completely agree about keeping an open mind. And I guess one thing that upon starting as a researcher on the bench that I had to learn is that research is really fickle and things are not always going to go the way that you want to. And you have to be really flexible about accepting that your experiment failed and you need to start over. And that happened a lot on the bench, and it's kind of the reason why I wanted to leave the bench, because it was really frustrating and challenging. But it happens still on occasion with like the projects we do now, where we'll be working on something and this is just not going anywhere, and we'll just drop it and change directions. And it depends on funding too. So sometimes you'll have the best ideas in the world and you can't get them funded, and it's hard to move the project forward without the funding. So you really do have to be flexible, and like Adriana said, the project or the job can change in ways. But on the other hand, one thing that's nice, I feel like I've had projects where I'm like, I really do not like this project, but then it goes away and something else gets started. So it's not always bad. So yeah, just a lot of flexibility though.
Any final or closing thoughts you want to make sure our attendees here? Well, I would like to thank you all. Oh, Adriana, I saw you come off mute.
I was just gonna say, like, honestly, like what we do is pretty fun. So I definitely encourage you to take a step into at least figuring out like, oh, let me try a few things out. Like I was saying, like whether it is like, some educational kind of webinars, if it's really like the beginning of what you're trying out, or going in through a hackathon, or talking with a professional, do at least take that step, because it can be actually pretty rewarding. And like we're talking a lot, like a lot of what we're saying might sound kind of technical, but then once you actually talk with someone that uses the website and they're like, oh, thank goodness you exist, like, oh, like some help, because it's kind of impossible to know everything in medical genetics, and you're like, wow, you actually have an impact. Sometimes you might lose kind of, like sides of the end goal, because you're like so into your research project, or so, like into, a particular project, that you're not like really thinking like, yeah, but actually I am contributing to a bigger goal that someone is going to benefit, and it's actually going to be good. And don't get so boggled up on details that you don't see the impact of what you're doing. And it is kind of fun. So I definitely encourage you to explore this as a potential career, because it is pretty cool.
Excellent. Thank you all. For our attendees, I just want to put up a reminder that we do have another session next week, our final session. There is a question about the difference between a biocurator and a variant scientist that I don't think we'll have time to cover, but I would encourage that person to reach out by email and we will get you an answer to that question. And for anyone who missed part of today or any of the other sessions, right on our homepage, clinicalgenome.org, you can find a link to all of the recordings that we have so far. And with that, we will be sending out a post-series survey afterwards. We'd love your feedback to help us put this panel on next year. So thank everyone for attending. Thank you to all of our panelists for sharing your experiences. And hope you all have a good rest of your day.
Thank you so much for
Thank
having
you.
us.
Yeah, thank you.
Have a good